In order to be considered living, you must follow the criteria for life, which includes reproduction, order, growth and development, response to environment, energy processing, evolutionary adaptation, and regulation. Let's take a look into biology, starting at the molecular level. Before we begin the story of the life of carbon, it is important to understand the structure of carbon. A carbon molecule is made up of 6 atoms and is able to form covalent bonds with other molecules due to its 4-electron outer shell. An example of this is carbon dioxide. It is also important to know that carbon can be found in 4 different macromolecules. The first macromolecule is the carbohydrate, composed of carbon, hydrogen, and oxygen. Bread and its high glucose content are an example of carbohydrates. The second is lipids, also composed of carbon, hydrogen, and oxygen. Lipids can be saturated or unsaturated depending on their fatty acid chains. This diagram shows an unsaturated lipid. Avocados are an example of lipids. The third is proteins, made up of carbon, hydrogen, oxygen, and nitrogen. All animal meats are an example of protein. The fourth and final macromolecule is nucleic acids, made up of carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Nucleic acids make up most DNA, and ATP is an important one of them used in cellular respiration. Now we can begin the life of a carbon molecule. In this film, we will be starting at the site of an erupting volcano. As the volcano erupts, carbon enters the atmosphere in the form of carbon dioxide. This carbon dioxide can now enter into the leaf of a plant through the stomata and into the chloroplasts. Now, inside the chloroplasts, this carbon can be used in the process of photosynthesis to help power the light-independent reaction known as the Calvin cycle. Carbon enters this cycle and exits as a part of the sugar known as glucose. Glucose is found in many foods, including this burger here. When we eat and digest this burger, our body is able to use its glucose to power the process of cellular respiration. The first step to cellular respiration is glycolysis, which converts glucose into 2 pyruvates and produces 2 ATP. The carbon then undergoes the bridge reactions and enters the Krebs cycle. It is important to note that the Krebs cycle occurs twice for every glucose or once for every pyruvate. Among the many things produced is carbon dioxide, which exits the cycle and is no longer needed for cellular respiration. The final step still needs to occur, however, known as the electron transport chain. The carbon dioxide that is released from the Krebs cycle leaves our body when we exhale and returns to the atmosphere. As carbon is able to leave our cells through the carbon cycle, how does oxygen enter our cells? The answer is cell transport. Cell Transport by Sammy and Judah.
According to cell theory, all living creatures are made up of cells, and these cells each have a number of intricate processes and systems that keep themselves and larger creatures alive. These processes range from converting sunlight into energy to storing genetic code or regulating what moves through the cell. And they all play an important role in keeping the systems of plants and animals working properly. In this video, we'll look specifically at the cell membrane and all it does to help the cell.
Cell membrane is a semi-permeable organelle that surrounds the cytoplasm and encapsulates the cell. The cell membrane is a phospholipid bilayer with integral and peripheral proteins along with cholesterol. The cell membrane is selectively permeable because it allows some molecules to pass through more easily than others. Now we'll go down to the cellular and molecular level to take a look at some of these intriguing interactions.
The first process being shown is simple diffusion of oxygen into a cell. This is an important process in helping the organism because it allows substances to move in and out of the cell depending on the concentration gradient. Simple diffusion does not require any proteins or ATP because it follows the concentration gradient and generally moves toward nonpolar and/or smaller molecules like water and oxygen. This is a form of passive transport.
The next process being shown is facilitated diffusion of glucose. This process is important because it allows ions and nonpolar molecules to enter the cell that are unable to enter through simple diffusion. Facilitated diffusion uses proteins to transport these polar molecules and ions into and out of the cell. No ATP is required for facilitated diffusion to occur, meaning that it is a form of passive transport.
The final process being shown is transport through the use of a sodium-potassium pump. This is an important process because it pumps ions into and out of the cell in a 3:2 ratio, 3 sodium ions for every 2 potassium ions. The sodium-potassium pump is a protein that moves sodium and potassium ions against the concentration gradient and utilizes ATP to work. This means that it is a form of active transport.
Just as proteins aid in cell transport across the membrane, they also drive the process of DNA replication in the nucleus. DNA replication by Sami and Judah. Before we begin, it is necessary to understand the cell cycle and which steps of DNA replication will be occurring. The first stage of the cell cycle is gap 1. Synthesis comes next, then gap 2, and finally mitosis. For this video, we will be looking only at DNA replication in synthesis. Here is DNA, made up of nucleotides, which each include a phosphate group, a deoxyribose sugar, and a nitrogenous base. Now, let's meet the cast, or the important enzymes used in DNA replication. The first enzyme is helicase. Helicase unzips or separates the 2 strands of DNA by breaking the hydrogen bonds between their bases. The next enzyme is primase. Primase lays down RNA primer, which is used to direct polymerase on where to build. Polymerase, the third enzyme, builds new strands of DNA by reading the primase 3' to 5' and building in the 5' to 3' direction. We'll go over 3' and 5' later. The final enzyme is ligase, which fixes Okazaki fragments and glues together uneven DNA strands. Going back to 5' and 3', these classifications are determined by the deoxyribose sugars on each strand. The strands are labeled 3' to 5' because of the carbon orientation of the deoxyribose sugars. These directions affect the way that the enzymes will travel through each strand. The first step of DNA replication is conducted by helicase, which runs through the hydrogen bonds between the nitrogenous bases and opens up or separates the strands. The second step is conducted by primase, which lays down RNA primer in the 3' to 5' direction. The third step is conducted by the polymerase enzyme, which reads the primase 3' to 5' and lays the daughter strands 5' to 3'. The fourth and final step is conducted by the ligase, which will fix the Okazaki fragments and glue together uneven DNA strands. Our DNA has now been replicated. Each new DNA consists of 1 parent strand and 1 daughter strand. It is important to note that the parent and daughter strands both have 5' and 3' classifications. Thanks for watching. As we complete DNA replication during synthesis of the cell cycle, we continue through gap 2 until reaching the process of mitosis, where cell division occurs. For every living creature to be able to grow, certain processes must take place within the cells to replicate the DNA and organelles. This makes sure that the full creature can live and work successfully, and the special name for this group of processes is mitosis.
The processes are divided into 8 parts, called phases. The first 3 phases are grouped as interphase, and the cell spends the most time in these while it performs its normal duties and replicates its organelles. The cell will go through various checkpoints and activities throughout its phases, and during synthesis, it replicates its DNA.
M-phase. This is the most important part of cell replication, where the nucleus and DNA split. It begins with prophase, where the nucleus begins to dissolve and the chromatids pair together. Each pair of identical sister chromatids is called a chromosome, and they are exposed inside of the cell at the end of prophase. The spindle fibers also begin to sprout out of the centrioles on either side of the cell. which will help to divide the chromosomes equally.
Next comes metaphase. In metaphase, the spindle fibers attach to the chromosomes at their centromeres, and then the chromosomes line up in the center of the cell. After the chromosomes line up, the anaphase stage takes place. In anaphase, the chromosomes are pulled apart and the individual chromatids move to either side of the cell as the spindle fibers retract.
Once the chromosomes have been brought to either side of the cell, telophase occurs. During telophase, two nuclei begin to form around the separate groups of chromosomes as the spindle fibers dissolve. The cleavage furrow also forms around the center of the cell and begins to pinch the cell membrane inwards.
With the pinching of the membrane initiated, the final phase of cytokinesis begins. In cytokinesis, which literally means cutting of the cytoplasm, the cell divides into two complete daughter cells. The cell membrane is pinched where the cleavage furrow was, and the two nuclei fully develop in each cell, with the chromosomes inside them unraveling into loose DNA. Finally, two complete cells are made and mitosis is complete.
Just as how DNA strands are necessary for mitosis to occur, they are also used for the creation of proteins. Let's take a look. Protein synthesis, transcription, and translation. The first step in the process of protein synthesis is transcription, which can be broken down into three parts: initiation, elongation, and termination. The first step, initiation, begins inside the nucleus of a cell on a loose strand of DNA. For the example within this video, we will demonstrate with a gene called MCR1. Several small proteins called transcription factors attach to a point on the strand and chemically signal the DNA where to begin unwinding. They signal where the entire process of transcription should take place.
Next, the large protein RNA polymerase binds onto the strand of DNA and begins separating or unzipping the two DNA strands from each other. Simultaneously, it lays down new messenger RNA, also called mRNA, nucleotides that correspond to one side of the DNA strand. This part of transcription is called elongation. The mRNA copy being made pairs to the template strand and is opposite as well as identical to the coding strand.
Next comes the step of termination, when the mRNA chain is meant to stop. The RNA polymerase needs to be signaled by the DNA where to finish elongation. A small 3-nucleotide sequence called the stop codon lets the RNA polymerase protein know when to end.
The now finished mRNA strand needs to exit the nucleus and be prepared for translation. The nucleotides are edited and refined. Then the mRNA receives a small buffer of nucleotides on either end to protect it before being sent through the cytoplasm to a ribosome. At the ribosome, the process of translation begins. Now, inside the ribosome, the mRNA enters in between the large and small ribosomal subunits and is ready to be read and translated. There are 3 chambers within the ribosome known as the E, P, and A sites. The mRNA is read in small groups of 3 nucleotides, and these small letter groups are called codons. Each codon passes into one of the sites at a time, the ribosome reading in the order of A, P, and then E. As each codon initially enters the ribosome, mRNA codons are read and matched to amino acids. These amino acids, connected to and carried by transfer RNA, will build the protein as the translation process is initiated.
As the mRNA continues to move through the ribosome in groups of 3 nucleotides, the amino acid chain lengthens during elongation. Each tRNA molecule corresponds to an amino acid, which is coded for by a specific mRNA sequence. The tRNA has its own anticodon that helps it find and pair to the correct mRNA codons at the A site. As mRNA moves through the ribosome, the amino acids connect to each other and lengthen over time, eventually completing a polypeptide chain.
This polypeptide chain, when finished with a combination of different amino acids in the body, is terminated with a stop codon similar to transcription, and then slowly folded into a protein. The folding process is delicate and precise, but results in a finished functional protein with the correct shape to perform a job. In this case, our finished protein is the MCR1 protein that will determine the fur color of a mouse.
The specific sequence of amino acids from these proteins helps give us our traits that make us distinct from one another. This phenomenon is known as genetics. Genetics, by Judah and Sammy. Here we have 2 flowers of the same species, but in 2 different colors, purple and white. What causes this difference in appearance within the smallest kind of groups? The answer is alleles. In every living organism, DNA within the cells codes for physical characteristics, and alleles are the specific variations in DNA within organisms of the same species. These can be any sort of difference from flower or fur color to the size and height of a creature. and are represented in their scientific studies by capital and lowercase letters. This letter is inconsequential but useful for differentiating alleles. So for this example, let's use the letter D. Depending on the relationship that the alleles have to one another, they can be recessive and dominant, with the dominant trait masking the recessive, along with other variations such as incomplete dominance, codominance, and polygenic traits.
For this example, once again, we will use a simple recessive and dominant relationship. The capital letter in genetics always symbolizes the dominant allele, and the lowercase symbolizes the recessive. So the trait for purple flowers is dominant, while the white trait is recessive. We can model the breeding and mixture of these alleles and the descendants of organisms with a Punnett square. The Punnett square is a tool in genetics used to predict the outcome of breeding two parents and what traits their children will inherit, displayed in a table based off knowledge of the recessive and dominant alleles. When two organisms are crossed in a Punnett square, the parents' two genes are written on the top and left side of the square, with one allele for each row or column. Every organism possesses two alleles for every trait, because they receive one from each parent, and which allows for the dominant and recessive relationships to occur within a single creature.
To complete the Punnett square, the table is filled out similarly to a multiplication table, where the alleles that intersect vertically and horizontally are paired together. In a simple monohybrid cross, shown in this Punnett square, there are always 4 outcomes for the next generation, but this can grow larger with a cross of multiple genes in a larger table. For our monohybrid cross scenario, the 4 offspring that can be made are all purple, because each only has the possibility to inherit one recessive and one dominant allele. This specific genotype, or combination of 2 alleles, is called a heterozygous genotype, when an organism inherits both one dominant and one recessive allele. A homozygous genotype is when an organism has both recessive or both dominant alleles. However, for all our heterozygous offspring, the flowers will end up purple because of the dominance of that trait. Phenotype, or the visible expression of the genotype in an animal, occurs based on the homozygous or heterozygous combination. And for our 4 offspring flowers, their phenotype possibility is 100% purple.
In the MCR1 gene of a rock pocket mouse, there are some variations in the alleles for fur color. We can use a Punnett square to once again model the breeding and outcomes of multiple mice. For our example, we will show a cross between a heterozygous mouse, which is brown, and a homozygous recessive mouse, which is white. The brown allele is dominant in this scenario. As we complete the chart, their offspring will have the 2 following possibilities for genotype: homozygous recessive and a white coloring, or heterozygous with a brown coloring, both equal in possibility. However, just because a trait is dominant does not mean it will always be more common in the population. As shown, crosses that involve the dominant allele do not always end up completely dominant. And crossing a recessive mouse with another recessive mouse always ensures a recessive outcome. And due to natural selection, in many real-life cases, the recessive allele is far more widespread among species.
Finally, we will look at another way scientists organize a hypothesis or information about the way traits interact with one another. These charts, called pedigrees, can be used to look at genetic traits from ancestors within human families, as well as animals and plants, and can help doctors to locate dangerous inherited disorders. However, we return to the rock pocket mice for our model. We will use the same parents as previously from the Punnett square cross and show a hypothetical scenario for the outcome if these mice were bred and produced 10 offspring. The first generation of rock pocket mice is called the parent generation and is labeled as generation 1. The second group, or first offspring generation, is labeled with a 2. The squares symbolize a male and the circles symbolize females. The results of female or male are separate to the trait for fur color and were randomly selected for this hypothesis, but in real life are controlled by the 2 X and Y chromosomes. Similarly to the Punnett square, the offspring generation ended up with a 50-50 ratio, or 1-to-1 ratio, genotype for brown versus white coloring. Now that we know how genetics work, let's see how these genetic traits can change over time and be affected by their environment, with evolution and ecology.
Evolution and Ecology by Judah and Sammy. Finally, we will take a look at the culmination of how all the different systems and processes of biology and life interact with evolution and ecology. Let's say we have a population of mice that are white in color due to their homozygous recessive genotype. The trait for their fur will be modeled with the letter A. When these 2 white mice breed, there is a 100% chance that their offspring will also show the white trait. However, when a genetic mutation occurs, new alleles are introduced, which allows for new genome and phenotypes. In this scenario, the new allele can be represented by a capital A, which codes for the black fur color. Genetic mutations randomly occur and can be caused by various factors, but are extremely rare and cannot be manipulated or controlled.
In our rock pocket population, the white mice had adapted over time to survive in their environment by camouflaging with the light sand that matched their fur color. If a mutation were to emerge in this population and cause the mouse to have a black phenotype, it would be at a major disadvantage without the ability to hide, and it would almost immediately be selected against. In this habitat, the mutation for different color could easily arise, but because it causes lower fitness, the white trait will remain the majority of the population. Yet, when the environment changes suddenly or over time, new evolution will occur. In our scenario, a volcano that erupted and caused major lava flow in the white desert area resulted in the creation of an environment with lots of dark rock. This new habitat changed the fitness of both white mice and black mice because it gave the black mice an opportunity to camouflage and made the white mice stand out more. The white mice that somehow entered or were born in the lava rock area were hunted and the trait exited that area. However, in some smaller places where white sand still covered the environment, the white trait remains.
To display the alleles within the entire population, we can use a graphic to show the gene pool or the collective percentages of genes within the whole rock pocket mouse group. Before the change in environment, the gene pool only contained white mice and was the 100% recessive genotype and white phenotype. Genetic mutations would occur, but would quickly disappear due to natural selection. Yet, once the new darker environment was introduced and a mutation randomly occurred again, the dominant black allele became more and more widespread as it was more advantageous for survival. The gene pool depicted slowly changed over time to favor the black allele more, giving an example of basic evolution. At its simplest, evolution is simply change in the allele frequency of a population over time, caused by environmental factors and natural selection, which is exactly what happened to these mice. Next, to look at an example of this on an even larger scale, we shall dive into ecology, or the study of organisms' relationships to one another and their surroundings. This can be examined on any scale from a single population to a community of different species to even the entire biosphere of the world. Yet again, we return to the example of the rock pocket mouse for our model.
In our example, we show the environment after it has changed from the volcano eruption and with a strong growing population of black mice. Many predators exist in this habitat and all cause natural selection for every mouse. To look at one specifically, the hawk is a dangerous predator to the rock pocket mouse and relies on being able to spot mice from far away. Due to the camouflage abilities of the black mouse, this hawk puts selective pressure on the population of white mice that live in the dark rock area. It eliminates them more easily than the black mice, and in doing so, hunts the trait to extinction in that area and changes the frequency of the black allele. This is a perfect example of evolution, as the white trait is slowly selected, again, over time.
We can display this relationship between prey and predator in a pair of graphs displaying a before and after change in the environment. In 2000, the population of mice was much higher, but once they lost the advantage of camouflage, the hawk population very quickly grew over the next 10 years. Thank you for watching.
As shown through this video, all of these biological concepts are interconnected and play into the biological levels of organization. They build off of one another and are necessary for the others to occur. Thank you.